Hello, hi, my name is Guojun Shen. I work at IRCMS. Our lab is mainly interested in something called developmental morphogenesis, and we use the context of three biological systems to study developmental morphogenesis. So the context we study developmental morphogenesis, the EMT and gastrulation and mesoderm formation. So I will briefly explain why we are interested in these sort of things. So EMT stands for epithelial to mesenchymal transition. Because we are a multicellular system, and our body is not made of one cell, but about 30 to 40 trillion of cells in a way. So the cells in our body are organized as either an epithelial or mesenchymal structure. So the transition of epithelial to mesenchymal transition, meaning that the cells are very dynamic, the organization is very dynamic. Cells can be organized as epithelial, or they can be organized as mesenchymal structure. So during development, the tissue organization changes very dynamically during tissue formation, organ formation. And understanding the regulation of this process is very important. And more important than this is that in adult homeostasis, right? So abnormalities in EMT actually causes cancer. So majority, over 90% of the cancer concerns the structure which are normally organized in epithelial tissue. So something goes wrong with the cell proliferation regulation and something goes wrong with the EMT. So what happens is that local metastasis and distal metastasis is going to lead to fatality in cancer. So majority of the cancer problems are caused by problems not so much in the cell proliferation, but in terms of epithelial to mesenchymal transition. So we think that by understanding what's going on in normal development, we can have a pretty good idea of why, in the case of cancer, abnormalities in epithelial to mesenchymal transition leads to pathological phenomena in a way. The system we're using is something called gastrulation here. So gastrulation is a process in early development which initiates cell differentiation and give the spatial coordinates to cells in the multicellular system. A pretty nice drawing by Conrad Waddington many years ago depicts the phenomena of a cell which is pluripotent in the beginning and then roll down the landscape, a hill-like structure, and progressively restrict their differentiation fate. So gastrulation is the very beginning of this process, and we are studying what is regulating this. And this has something to do with the epithelial to mesenchymal transition as well. So for example, this is the human embryo here. And human embryo in the context of maternal uteral structure here. And if you magnify this area, the tissue which later on will give rise to you and I in every single cell in your body is actually a little structure here called epiblast here. And all the rest is something called extraembryonic tissues. So the epiblast will undergo a process called epithelial to mesenchymal transition I just mentioned, and they give rise to three different types of tissues called ectoderm, mesoderm, and endoderm cells. That's the very beginning of the cellular lineage differentiation, and we're trying to understand how EMT is playing a role in this structure. The second system we're using is something called extraembryonic mesoderm, which is very interesting in the sense that we're focusing on the mesoderm, and we know that in the human case and also in the chicken case, that the bird that we are studying, actually the vast majority of the tissue in the embryo is not the intraembryonic part, but the extraembryonic part, meaning that the embryo has to prepare its way to interact with the environment so that the embryo itself can get sufficient nutrition, get sufficient oxygen, etc., and dispose of waste. So these functions are performed by something called extraembryonic tissues, and the extraembryonic mesoderm is one component of this extraembryonic tissue. So we're studying how EMT is playing a role in this process. So we hope that by understanding these processes, we can actually understand how, in adult cases, human cancer can be somewhat slowed down if we understand how metastasis happens. And also for human congenital diseases, oftentimes it's caused by diseases from the fetal-maternal abnormalities in fetal-maternal interaction. So by understanding this process, how extraembryonic mesoderm is playing a role in this process, we should be able to understand how actually a majority of childbirth problems was actually caused by male interaction, the missing interaction between fetus and the maternal tissues. So that's our hope. And overall, about IRCMS, it's a very Kumamoto is a very nice city. It's not too big. It's not small. It's very close to nature, and its life is very convenient. So these things will not distract us from research. If you live in a big city like Tokyo, you probably have to commute more than 1 hour every day just to get to the workplace. Here, 5 minutes, 10 minutes bicycle ride, you will be able to get to your workplace here. And what drives me as a scientist to the research is that what I hope it can encourage a student is that there are so many wonderful things that we do not understand what's going on in life. And I think researchers' role is actually to uncover something, little something, which both satisfies our intellectual curiosity as well as benefit the society. And in this case, the satisfaction is to understand something which is not obvious. In our case, to connect the link between what's happening in the normal development and what is happening in the human disease cases, whether it's cancer or whether some other types of congenital disease. So other than research, what I do is I enjoy nature, and I also have a family, so I spend a lot of time with my daughter. My daughter is on the 2nd grade, and she's attending a local primary school, so I have to learn a little bit of Japanese to interact with the teacher as well. I do not speak Japanese, sorry about that. And the message finally to students who wants to study in Kumamoto in IRCMS, possibly in overall contact in Japan. Research in Japan is very good, and I think Kumamoto is very strong in both the developmental biology research, which is my main focus, as well as on stem cell-based research, which is the focus of a lot of other PIs in this institute. And I think you will have an opportunity, a very rare opportunity in Japan, that to a totally English environment, but very internationalized scientific interactions as well as personal life-wise interactions. And I am sure that if you come and take a look, if you have opportunities, or read some papers that are published by PIs from IRCMS, you will be able to get a fuller view of what's happening in IRCMS. So hopefully after the pandemic, we can see you in Kumamoto in IRCMS, enjoy science, enjoy life in Japan.